Hey everyone, my name is Ehsan. Welcome to our channel, Biomed Information. We are going to learn everything needed for cancer bioinformatics in these videos. In this session, we are going to cover the cancer biology. If you like the video content, please like, comment, share, and subscribe our channel. If you got any questions, please do not hesitate to ask in comment section or through my Gmail. I hope you enjoy the video. Hey everyone, welcome to our channel Biomed Information. My name is Ehsan and I am currently working on cancer vaccines. In these videos, we will try to learn everything needed for cancer bioinformatics. So we continue our journey on cancer biology. If you didn't watch the first episode of Intro to Cancer Biology videos, you can easily go and watch the first part. We have discussed the basics of cancer terminology and cancer hallmarks, as well as the differences between malignant and benign tumors. In this video, we will focus on causes and genes responsible for cancer development. There are three important processes that contribute to the overall net cell number in an individual. First, cell proliferation. We also can call it cell division or cell growth. So cell proliferation is the most obvious. Cell division results in two daughter cells. Second, the elimination of cells by programmed cell death also affects the net cell number. Third and last one, during the processes of differentiation, cells can enter an inactive phase of cell growth, and thereby differentiation can affect net cell numbers. DNA mutations that change the function of normal genes involved in growth, apoptosis, or differentiation can affect the balance of cell numbers in the body and leads to unregulated growth. So a change in the processes of growth, apoptosis, or differentiation can alter cell numbers. Normal genes that can be activated by mutation to be oncogenic are called proto-oncogenes. Proto-oncogenes play functional roles in normal cells. They are normal genes, but the term reminds us that all normal cells have genes that have the potential to be oncogenic. Growth is regulated by both positive and negative molecular factors. So, to increase growth, enhancement of positive factors or depletion of negative factors is required. There are two major types of mutated genes that contribute to carcinogenesis: oncogenes and tumor suppressor genes. A general description of an oncogene is a gene mutated such that its protein product is produced in higher quantities or has increased activity and therefore acts in a dominant manner to initiate tumor formation or development. On the other hand, tumor suppressor genes code for proteins that play a role in inhibiting both growth and tumor formation. Loss of growth inhibition occurs when mutations cause a loss of function of these genes. Consequently, growth is permitted. Tumor suppressor mutations are mainly recessive in nature because one intact allele is usually sufficient to inhibit growth. Therefore, both alleles of the gene must be mutated before the loss of function is actually seen phenotypically. All of the cancer cells in a patient arise from a single cell that contains an accumulation of initiation mutations. In other words, the development of cancer is clonal. It is generally assumed that only one of the 10 to the power of 14 cells in the body needs to be transformed in order to create a tumor. Environment, reproductive life, diet, and smoking are four major factors that are involved in carcinogenesis. These lifestyle factors can be altered or changed to prevent most cancers. Exposure to carcinogens, hormonal modifications influenced by, I don't know, childbirth or birth control, and exposure to viruses like HPV, which is involved in human cervical cancer, are totally underlie these lifestyle factors. The International Agency for Research on Cancer, or IARC, has classified materials in four different groups based on their potential to be carcinogen. Category 1 involves materials carcinogenic to human beings like sunlight and tobacco. 2A represents probable carcinogens to humans, like night shift work, while 2B discusses possible carcinogens, like coffee and lately classified aspartame in Diet Cokes. Materials in Category 3 are not carcinogens, and the things in Category 4 are possibly not carcinogen for human beings. There is a need to learn about the differences between normal and transformed cells at the molecular level in order to identify cancer-specific molecular targets. In this way, we can design drugs that will be specific for cancer cells, have increased efficiency, and cause fewer side effects. As we examine the molecular pathways that underlie carcinogenesis, we must keep in mind that the pathways do not act in isolation, but are interconnected as you see in the picture. Despite the many hundreds of molecules involved in carcinogenesis, there are several families of star players in the story of carcinogenesis. Many of these star players act as nodes that receive signals from many pathways and can exert several effects in response to specific signal. These star players are also called hub genes or hub proteins, which we are going to use a lot in bioinformatics classes since they are valuable targets. The family of protein kinases, one of the largest families of genes in eukaryotes, must be included in any introduction to cancer biology. Protein kinases phosphorylate, or in other terms, add a phosphate group to a hydroxyl group on specific amino acids in proteins. The Ras family is another star set of genes which are found mutated in over half of certain cancers, like colon cancer. Ras is an intracellular transducer protein that acts subsequently to binding of a growth factor to its receptor and is involved in transmitting the signal from the receptor through the cell. The other protein is tumor protein p53. Tumor protein p53 and its related family members like p21 uh, hold essential positions in guarding the integrity of the genome by coordinating responses of the cell like cell cycle arrest, DNA repair, apoptosis to different types of stress like DNA damage, hypoxia. The p53 gene is a tumor suppressor gene that has a key role in inhibiting carcinogenesis, and therefore it is called the guardian of the genome. It is mutated in more than half of all cancers, and over 1,000 different mutations have been identified till now. It acts as a transcription factor and induces the expression of genes required to carry out its function. The last gene we are going to discuss in this introduction is the RB, the retinoblastoma gene. The retinoblastoma gene is also a tumor suppressor gene that plays a central role in regulating the cell cycle. It is commonly mutated in several cancers. The retinoblastoma protein normally functions as an inhibitor of cell proliferation by binding to and suppressing an essential transcription factor of cell cycle progression. Its activity is regulated by phosphorylation by cyclin D and the cyclin-dependent kinases. If you want to discuss hub genes in detail, please write down in comment section and we will try to make educational videos for these stuffs if you like these content. So I think we have enough biology information. Never enough. I'm gonna assure you it is never enough. But I think it's enough to start the cancer bioinformatics. If you like the videos and want to learn more about cancer biology, the pure cancer biology, please write it down in comment section to encourage us to make more videos about cancer biology. If you like the video content, please like, share, and subscribe our channel. If you got any questions, please do not hesitate to ask in comment section or through my Gmail. I hope you enjoyed the video.